Hi, welcome in this session on AI and diagnostics. My name is Olivier Givard. I'm the moderator of this session, and I'm an associate professor at Stanford at the Department of Medicine and the Department of Biomedical Data Science. And I will start with the first talk in this session, followed by Dr. Kim and then Dr. Chan. So I'm going to talk about biomedical data fusion for cancer diagnostics and prognostics. So I hope I don't have to convince you that machine learning has been successfully used for biomedical data analysis for several heterogeneous data types. And with biomedical data types, what I mean with that is different types of data, such as clinical data, molecular data, and imaging data. And many studies have shown that biomarker discovery from these biomedical data contributes to precision medicine. However, before I continue, I need to define what is precision medicine. And with precision medicine, I mean the optimization of treatments of specific patients or subsets of patients using biomarkers. And original work in this area has been mostly targeted at discovery of molecular biomarkers, including genomic and transcriptomic biomarkers. I'm listing here a few examples of molecular biomarkers that are currently in use in clinical practice. And the goal of these biomarkers is to accurately assign diagnosis or treatments to patients. But more work is needed, and biomarkers are needed for accurate diagnosis and treatment assessment. For example, in glioma, there is a need to distinguish pseudoprogression versus real progression. And there's, of course, many other examples. But one point I want to make is that molecular biomarkers are not always enough. And there's other data sources that we can consider, including clinical and image data. And these, especially imaging data, has been made available for analysis thanks to advances in quantitative image analysis and deep learning. And I'm talking here then specifically about whole slide images in pathology and radiographic images in radiology. And there is a need to develop models that can efficiently integrate these different types of data and also combine multi-scale data. So what do I mean with multi-scale data? I mean data that characterize molecules, cells, tissues, and patients. For molecules, we can have DNA and RNA sequencing technologies that generate molecular data. In terms of cells, we have whole slide images, immunohistochemistry or codecs, or any type of digital pathology images. And at the tissue level, we can have radiographic images that give us data at the tissue level. The reason for integrating all these different types of data is as follows. Essentially, we argued that multi-scale technologies sample heterogeneity in different ways. For molecular data, we get a deep detail of one piece of tissue, or we get cellular heterogeneity, for example, or we even get detail of a single cell. With immunohistochemistry, we get a slice of tissue, and we know the cellular architecture and, for example, infiltration of immune cells. And with radiographic image data, we get 3D information of a whole organ or a tissue as a whole. And so there's a huge opportunity to study synergy between all these different scales. One example of multi-scale data fusion is the field of radiogenomics or imaging genomics, where you essentially can link radiographic images or medical images with molecular data. For example, computational analysis of histopathology images using morphometric features and deep learning, and associating that with molecular data, including genome-wide gene expression data or copy number data or DNA methylation data. Today, I'm going to give two examples of integrating imaging and molecular data. The first one is linking DNA methylation signatures with histopathology images. And the second one is an example where we use multimodal data fusion in brain tumor research. First, I'm going to talk about the DNA methylation patterns reflected in histopathology images. To briefly summarize or give you a background on DNA methylation, this is the transfer of a metal group to a CG dinucleotide. And essentially, what this does is, if you methylate a CpG site, essentially this turns off gene expression. So this causes gene expression silencing. And this is a common mechanism in cancer, which with hypermethylation of genes and hypomethylation of genes. Hypermethylation essentially inactivates genes, and hypomethylation can activate genes. And this is an important process in oncology, where there's these are alternative mechanisms to activate or deactivate genes as an alternative mechanism to copy number alterations and somatic mutation. For example, MGMT methylation, glioblastoma, and BRCA1 methylation in ovarian and breast cancer are common examples. Other observations that have been made in cancer is the SIM phenotype, which is the CpG island methylator phenotype, which is characterized by hypermethylation. For example, this exists in SIMP and is caused by a mutation in BRAF. There's also glioma SIMP subgroup, which is correlated with IDH1 mutations, and similarly in acute myeloid leukemia. So we previously developed an algorithm called MethyLmix to identify DNA methylation-driven genes in cancer. And now what we want to do is we want to see what the relationship is between methylation patterns and computational pathology. In computational pathology images, we get a picture of the cellular architecture of human tumors. And diagnostic information is embedded in these images. But huge resolution of these images often prohibits manual interpretation. And but we get complementary information from these images in terms of cellular heterogeneity, cellular structure, and cell type composition. So here's the workflow of this study. Essentially, we want to link image features that we extract from these digital pathology images with DNA methylation changes that we process using the MethyLmix algorithm. So here's the data that we used for this project. We used about 1,000 patients from the Cancer Genome Atlas, including glioblastoma and low-grade glioma. Then we processed the molecular data of this data set using MethyLmix, including tumor and normal DNA methylation data. And we also integrate RNA sequencing data. Using MethyLmix, we are able to identify about 900 hyper and hypomethylated genes. So this is essentially the pre-processing we did on the molecular data. Next, for the same set of patients, we also have whole slide images, which are stored on the GDC portal, the Genomic Data Commons portal from TCGA. From these images, we extracted morphometric features, and these are essentially image features or texture features that capture the cellular composition and heterogeneity from these tissues and from these patients. And these are cellular morphometric features and 8 contextual features. And they are summarized using first-order statistics, including mean, median, standard deviation, and so on. So once we have these image features and also these MethyLmix states, we can essentially integrate these 2 types of data. And these are the 3 things that we investigated here. We first tried to predict single gene methylation states from image features. Then we also will I will show you how we clustered MethyLmix states to predict clusters, and we used 6 common classifiers, including random forest and support vector machines. Here are some results. So first, we tried to predict the methylation state of single genes using these image features. And what we saw is that most classifiers essentially performed similarly, except naive Bayes, which was worse, and we essentially dropped then naive Bayes from the analysis. And the average area on the ROC curve, which is the performance metric that we use to evaluate these models, is 0.7. So here are some examples of genes that we could predict for glioma patients. So CDK4 and MICBP are genes where we could predict the methylation status from these image features. And both of these genes are involved in cell cycle progression. Next, we also wanted to study what happens when we look at groups of methylation patterns, because we didn't expect that each single gene's methylation status we could actually predict from these image features. So we essentially clustered the MethyLmix genes into subgroups, and we found that 5 subgroups accurately represented these glioma. And next, then what we tried is we looked how we could predict these 5 subgroups using the morphometric image features from the digital pathology images. And what we found was that we had a much higher performance at the cluster level. So for these 5 clusters, each cluster we could predict with an area on the ROC curve of higher than 0.8, except for cluster 2, which had a worse performance, as you can see here on the left. So this is just an example of how whole slide images essentially reflect DNA methylation patterns of human tumors. And to summarize this part, essentially what we showed here is that morphometric features extracted from histopathology images predict both single gene methylation states and methylation clusters. And I showed you examples of these for glioma patients. We also tested this on another cancer site for renal cell carcinomas. And in principle, what we showed is that there's a relationship between epigenomic patterns and cellular representation of glioma. And the interesting application of this first project is that image biomarkers exist that essentially reflect these molecular properties. Next, I wanted to show you another project where we used a data fusion framework using multi-scale data of adult and pediatric brain tumor patients. And here we are using this model for prognostics. So we are focusing on prediction of survival. Here's the data that we used in this project. So we had molecular data, we had tissue level data, cellular data, and clinical data. And the question here is that, can we use multi-scale data from adult and pediatric brain tumor patients to predict overall survival? So here are a few technical details about the cohorts that we used and the pre-processing that was done on these data. First, we had about 1,000 patients. This is the same cohort that I mentioned in the previous project of about 1,000 samples, where we had both histopathology and gene expression data. The gene expression data was for about 12,000 genes. And then we also used age and sex of these patients. And these are essentially the adult glioma. The second cohort that we used is a pediatric glioma cohort of about 200 patients, where we had the same data modalities available, namely histopathology and gene expression data. And again, we had about 12,000 genes, and we used the whole slide image data. And we had both age and sex again in terms of the clinical features. Here are a bit more details about the pre-processing. The image data was pre-processed as follows. First, we need to decide, or we need to detect the places where the tissue are in the images. So we used an Otsu segmentation mask to identify the foreground images from the background. And then, because these histopathology images are too big to model directly, we need to extract patches of 224 by 224 pixels from the foreground. And then finally, and also a common step that is done in modeling digital pathology data is data augmentation or data transformation, where we flip color jitter and we normalize these images to avoid overfitting. For the gene expression data, we used very standard pre-processing methods followed by log transformation and Z-score transformation. So here is an overview of the actual model that we used. So we have essentially a feature extraction module for both the image data. For the image data, we used ResNet 50. This is a convolutional neural network that is pre-trained on ImageNet on about 1 million photographs. And we retrained this model on the digital pathology data. Then for the gene expression data, we used multi-layer perceptron that can essentially model these 17,000 gene expression features. Then we have a Cox loss module that is then used to predict survival of these patients. So we are essentially predicting prognosis. So first, before we try to look at combining these different data modalities and predicting overall survival, we established the baseline performance of each of the different modalities. And this is what I'm showing you here. We are using 2 performance metrics in this work, the concordance index and the Brio score. The concordance index is similar to the error-under-drop curve, and an ideal concordance index is 1, and means that we can perfectly distinguish poor and good survival cases. It's important to mention that the concordance index is a relative metric and essentially scores how well we are able to rank patients in a test cohort. Secondly, we also use the Brio score. This is an error metric, which means that lower Brio scores are better. And this is also an absolute performance metric. As you can see, the RNA data only, so the gene expression data only had the best performance in the baseline model with a concordance index of 0.91 and a low Brio score of 0.08. And this is for the pediatric glioma data, and similarly for the adult glioma, we also saw that using just gene expression data had the best performance, with a concordance index of 0.78 and a Brio score of 0.15. So now let me introduce you the different types of fusion that we developed to combine these different types of data. And we tested essentially 3 different types of fusion: feature fusion, late fusion, and joint training fusion. Here is feature fusion. In this case, what we did is we built a separate model for each of the different data modalities. So we have ImageNet, or ResNet, I mean, for the image data, and we have a simple multilayer perceptron for the gene expression data. And then the features are combined, and then a simple multilayer perceptron is used to combine the different features that are extracted to predict then overall survival. Next, we also tested late fusion. In case of late fusion, we built a separate model on each of the different data modalities and then integrate the predictions. So each model essentially makes a prediction of the prognosis, which is visualized here. And then we just look at these 2 predictions and combine them using a very simple Cox regression model. And that gives us then a risk score. And finally, we also tested a joint fusion. This is the most complex model, where essentially a model is trained end-to-end to predict the risk score, and where this is the only model where parameters of each of the different modalities that can influence each other. So here are the results for the adult glioma cohort. So at the top, I still mention the performance of the single modalities for these adult glioma, and then at the bottom we have feature fusion, late fusion, and all the different fusion examples. As you can see, fusion did outperform each single modality with a concordance index of 0.79 and a lower Brio score. And this is a model for adult glioma. Similarly, for pediatric glioma, we were able to improve the performance for predicting overall survival of these pediatric brain tumor patients. And in this case, joint training fusion had the best results with a concordance index of 0.95 and a Brio score of 0.08. Here I'm also showing the survival curves for these 5 models, essentially showing that the 2 fusion models at the bottom provided the best result. So this is an alternative way of looking at how well these models are predicting survival by looking at a poor and a good prognosis group using Kaplan-Meier survival curves. Next, we also wanted to try if we could transfer these models between the different types of cohorts, so between adult and pediatric glioma. The main motivation to do this is because we have more adult glioma data than pediatric glioma data. And the question is, can we transfer models and can we improve the performance of these models using transfer learning? And we tried 2 different types of transfer learning. First, we tried if we build a model on the adult glioma and test it directly on the pediatric glioma versus a model that is additionally fine-tuned on a subset of pediatric glioma cases and then test it on the same remaining cases. And of course, we can also do the opposite from pediatric glioma to adult. Here are some results of this transfer learning experiment. And this is from adult to pediatric glioma. So without fine-tuning, when we build a model and we directly test it on the pediatric glioma, the model works okay, but it's not outperforming the single modalities. So we get a concordance index of 0.67 using feature fusion. However, with 50% fine-tuning, we are able to reach, again, the best performance with a concordance index of 0.95 and a Brier score of 0.09. It's important to mention whether, even though that this performance is the same as building a model directly on pediatric glioma, in this particular case, using transfer learning, we used much less data. Next, and finally, we also wanted to investigate whether we could also interpret this model, this data fusion model, and if we could highlight how the model is making decisions. For the image part of this model, we looked at saliency maps to see what areas of the image are being used to make predictions. This is the patient with the poorest survival, and it's a high-grade tumor with sarcomatous features. And these areas that are pointed out with these arrows are essentially areas of apoptotic cells and necrotic cells. And these are the areas that the model is using to make predictions. Similarly, here is a patient with good survival, and the arrows are pointing to areas with less cellularity. And so these are the features that the model is identifying to be involved in good survival. Secondly, we can also look at the gene expression values and the genes that are getting selected in this model and which ones are important for making predictions about survival. Instead of looking at single genes, we actually wanted to look at pathways because this provides an easier way of interpreting the results. And in this particular case, we used the reactome pathways to look at model interpretation. And what I'm showing you here are the feature importance of these pathways, where positive values mean that these pathways are important for poor prognosis, and negative values mean that they are important for good prognosis. And we found that important and interesting pathways are selected in this fusion model. We found, for example, that cell cycle and RAS signaling are important in poor prognosis, whereas RUNX3 pathways are important in good prognosis. And that was reassuring because RUNX3 has recently been reported as a tumor suppressor gene in glioma. It's also important to mention that when we looked at model interpretation of the baseline model without imaging data, so the model that's only based on RNA sequencing data, that we could not see these pathways, and the pathways were more randomly distributed. So to summarize this part, I hope I have shown you some examples where multiple modality data fusion can improve survival prediction from single data modalities. And I also hope that I have shown to you examples of transfer learning where survival models can be learned from one cohort and tested on another across age groups and tumor subtypes. More work needs to be done on interpretation of these models to understand better how they make decisions. Overall, our focus is to build machine learning models to integrate heterogeneous data, including molecular data, genomics and epigenomic data, with radiographic and digital pathology images, and using all these different data sources in a multimodal data fusion framework for diagnostics and prognostics. Multiscale modeling essentially shows how molecular biology properties are reflected in these images, in histopathology images and radiographic images of tumors, and how these can be used as surrogate biomarkers for both diagnostics and prognostics. And we argue that this is a promising application of non-invasive precision medicine. And also that these imaging features can be used as a biomarker for treatment decisions and also for treatment response. These are the people I have to acknowledge for this work. And I'm happy to answer any questions, which you can submit using Slido. So I see a question that was just submitted. So the question is, are you convinced that feature fusion will be superior to late fusion in most applications? I would not say that it's not possible to say beforehand that a particular type of fusion, for example, feature fusion, will be better than another type of fusion. This is very application dependent and really depends on the problem that you're working on. So also in the results that I just showed you, the results were the type of fusion was not the type of fusion that had the best result was not the same in the adult versus the pediatric glioma. So I would definitely not claim that any sort of feature fusion strategy will, or any data fusion strategy will outperform any other strategy. Another question was just submitted. How do you define positive labels and negative labels in the transfer learning? So for the particular example that we gave there, the label that we are trying to predict here is overall survival, right? So we're trying to predict the time that the prognosis of these patients. So there are no, this is essentially a survival problem. So this is more of a continuous value problem. So there are no positive and negative labels. There's survival time for these patients. So we have these patients for all these, for both pediatric and the adult patients. So we are able to use the label. So this is there was no problem of labeling these cases. I hope that answered your question. And maybe one more question before we move on from Adam Alessio. Do you know if there are methods to optimize where to fuse features from multiple modalities for a given application, or how to select fusion architectures? The methods to do these are very similar to methods when you have different types of models. So for model selection, we essentially can use cross-validation or randomizations where different training and test sets are being used to compare different fusion strategies. And then we have to look at performance metrics depending on the different type of outcome that we have, and essentially compare all these fusion strategies on unseen data. And this is very similar to optimizing any type of hyperparameter. So essentially, the fusion strategy then becomes a type of hyperparameter. Okay, let's move on to the next speaker, Dr. Daniel Kim. He is an assistant professor at UC Santa Cruz, an associate member also of the Canary Center at Stanford for early cancer detection, and his lab is mainly focused on RNA liquid biopsy technology. Take it away.
Okay, great. Thank you so much, Olivier, and to the organizers for this opportunity to share some of the work that my lab has been doing to really harness these millions of RNA features in the genome to advance human health. And just to kind of start off with, we're really excited about the potential for liquid biopsy to diagnose disease. And I'm sure many of you have heard of all of these companies that are developing these liquid biopsy assays to be able to enable cancer early detection and early detection of other diseases. But really the focus of a lot of these different platforms has been on looking at DNA as the analyte of choice. If you imagine, for example, in a cancer, when the cancer cells die, they release DNA into the circulation, and that DNA gets fragmented, it gets degraded. And so you end up having very small pieces of DNA, about 150 base pairs in length, that are circulating in the blood. And so the way that a lot of these companies are currently doing this is to identify these pieces of DNA that are released into the blood, to sequence them and to identify either mutations that are present within these pieces of DNA or changes, for example, in methylation. But we're really focused on the potential for RNA, especially for the early detection of disease, because we know that, for example, for early cancer detection, DNA as an analyte does not seem to be sufficient to enable highly sensitive early detection of cancer. But the nice thing about RNA is that instead of the cancer cells having to die and turn over to release the DNA, RNA is constantly being secreted by cells, cells that are alive, especially cancer cells, which tend to preferentially release RNA in these extracellular vesicles, such as exosomes. And so the RNA is packaged into this very pristine state. And when you can take some blood from a patient and isolate these vesicles, you can identify the RNA that's being secreted by these cancers. And so for specific types of cancers, I think Olivier had mentioned RAS as one of the pathways that he also sees. We've seen that the activation of these oncogenic signaling pathways tends to lead to hyper transcription of certain classes of RNA, which I'll be telling you about, and also the preferential secretion of these signals from the tumor. And so today, what I'll really be focusing on is using RNA as the analyte of choice to enable disease diagnosis, particularly focusing on cancer. And so when you look at a typical machine learning workflow for disease diagnosis, there are several kind of canonical steps in terms of data cleaning, featurization extraction, model fitting, and developing your classifier. But really today, I'll be focusing on one aspect of this machine learning workflow, particularly this aspect of featurization extraction and the role that RNA plays in identifying very prominent features that are upregulated, for example, at the earliest stages of disease, and how we can leverage these RNA features to really enable early detection of diseases such as cancer. And so just to give you a little bit of background regarding why we focused on RNA, when you look at the human genome, there are over 3 billion base pairs in the human genome. But what's really striking and quite surprising is that most of this 3 billion base pairs is transcribed into RNA. And so if you look at the 3 billion base pairs in the genome, only about 1 to 2% encodes proteins, while the vast majority of it is generating non-coding RNA, so RNA that does not get translated into protein. And so there is this very vast feature space that can be leveraged for early detection, but we haven't been doing that up to this point because we haven't really understood what are the right features to look at, what are the right types of RNAs that enable this early detection of disease such as cancer. And so when you think about, for example, the non-coding genome or the non-coding transcriptome, which is the RNA output of the genome, there are many different types of RNAs. And so if you look at just the human genome, this contains over 100,000 non-coding RNA features, which can be leveraged for disease diagnosis. And so here in this review by John Rin and Howard Chang, what they've shown is that over the past few decades, we've discovered many of these different classes of non-coding RNAs. For example, one kind of more recent class of non-coding RNA called long non-coding RNAs are in fact very similar to protein coding messenger RNAs, but they just don't make protein. And so if you look at, for example, just the number of long non-coding RNAs, just as one particular class out of these many different non-coding RNA classes, we could see that there are upwards of 58,000 of these long non-coding RNA genes within the human genome. And this number is only going to increase as we sequence more deeply different tissue types, different cell types with the Human Cell Atlas efforts, for example. And there are some estimates that suggest that there are over 100,000 of these long non-coding RNA genes in the human genome. And that kind of doesn't include all of these other, again, classes of non-coding RNAs that can be, again, leveraged for disease diagnosis. But the other thing that is really quite striking about the human genome is that when you look at the kind of feature space of the genome, there are millions of these repetitive sequences called transposable elements. And so there are over 1 million of these transposable element features that can, again, be leveraged for diagnosis of disease. And if you look at some of the kind of more prevalent transposable elements within the human genome, for example, line elements or sign elements, what's really quite interesting is that in the context of healthy individuals or healthy cells, you normally don't see transcription of these transposable elements. So you don't see RNA that's being expressed from these transposons. But in the context of diseases such as cancer or Alzheimer's disease or other types of diseases that we've looked at, there is this aberrant upregulation of these RNAs that are being transcribed from not just, for example, link RNAs, but also these transposable elements and other classes of RNAs. And so this gives us a much, again, a richer feature space with which to develop these machine learning-based approaches for disease diagnosis. And so today I'll be telling you about some of the work that we've been doing to identify these transposable element and non-coding RNA signals that we can use to identify the best features for diagnosing disease. And so really the question is, can we leverage this vast non-coding RNA feature space to advance human health? And one of the ways that we're trying to do that is by doing cancer early detection, being able to detect disease before it's spread and metastasized to other parts of the body, because we know that for most patients that do get this cancer diagnosis, they die because the cancer has spread to other parts of the body as it's metastasized. And so if one is able to detect this disease at the earliest stages, for example, at stage 1, when the cancer is still localized within the body, this will lead to a much higher number of patients surviving and receiving a potentially curative treatment for this diagnosis. And so initially, we focused on KRAS because we know that RAS genes are very prevalent across many different types of human cancers. And at least for some of the cancers that we've looked at, we know that KRAS mutations are involved in the very earliest stages of cancer initiation. So if you look at, for example, pancreatic cancer or certain classes of lung cancer, we know that they're really driven initially by this gain of function mutation in KRAS that leads to this hyperactive RAS signaling, this increased proliferation. And for example, if you look at pancreatic cancers, we know that, again, this is a very early mutation that leads to the formation of cancer. And so what we wanted to do is really identify, when you have this very early mutation, how does this really regulate this vast non-coding RNA feature space in the human genome? And how can we, again, use this information regarding the earliest changes, the earliest molecular changes, to be able to diagnose diseases such as cancer at the very earliest stages? And so we used a very simple system to identify these early signatures. So we took some human lung airway epithelial cells, and we simply introduced a mutation in KRAS that's commonly found in lung cancers. And simply by introducing this one mutation into these cells, and this is just a single nucleotide change in a single gene, this leads to oncogenic transformation in this kind of in vitro model, where if you take these cells and you implant them in mice, they form tumors. But in the absence of just this one oncogene, they won't form tumors in vivo. And we perform various transcriptomic and epigenomic profiling assays to identify these RNA features or RNA signatures of this early mutation that really drives a lot of these cancers, with the goal to identify, again, the right types of RNA features that will enable the earliest diagnosis of disease. And so we were really quite surprised to see that at the very earliest stages of this transformation process, we saw a lot of interferon-related genes that were significantly upregulated by this mutation in KRAS. So here what I'm showing you is RNA sequencing data that's plotted in what's called a volcano plot. Here, this is just the fold change in terms of the differentially expressed interferon-stimulated genes. And so on the right are upregulated genes, and on the left are downregulated genes. And this is in the mutant KRAS cells relative to the control cells. You can see that all of these interferon-stimulated genes are being upregulated or differentially significantly upregulated. And so we wanted to identify, if what we were seeing in vitro was also relevant to patients in vivo. And so what we did was we took the Cancer Genome Atlas data, and Olivier had mentioned this in his talk as well, where we looked at all of the lung adenocarcinoma patients that had known mutations in KRAS. And so this is the particular mutation that we had studied in vitro. And so we looked at patients that had this same KRAS hyperactive mutation. And so here, each of these columns indicates a single patient's transcriptome for these particular genes that we had seen upregulated simply by introducing this mutation in KRAS. And what you can see on this heat map is that for patients that have this activating KRAS mutation, we see much higher levels of these interferon-stimulated genes in vivo, which recapitulates nicely what we saw in vitro as well. And so because we saw this, this very strong signature, we wanted to identify what are the RNAs that are potentially inducing this interferon-stimulated gene signature. And we wanted to look for transposable element expression, because we know from other studies in the field where people have, for example, seen aberrant upregulation of transposable elements in cancer due to treatment with, for example, DNA methyltransferase inhibitors, there is this subsequent upregulation of these interferon-stimulated genes, which sense these RNAs that are coming from these transposable elements as non-self. So there's this process termed viral mimicry, where the cancer cell thinks that it's infected by a virus due to these aberrant transcripts that are coming from these transposable elements, such as endogenous retroviral elements. And so when we looked at the transposable element landscape of these cells, again, when we looked at, for example, 2 different lung cells where we simply introduced a mutation in KRAS, what we see is that there is this upregulation of these transposable element RNAs. And so these are just some of the examples that we saw that were significantly differentially increased in these mutant KRAS lung cells. And so we believe that this is inducing this response in the interferon-stimulated gene pathway. But the other thing that was really quite striking is that because we saw this upregulation of these transposable elements, we wanted to see what's causing this upregulation, what could be responsible for this. And so we looked at another class of genes called Crab zinc finger genes. And so these are typically involved in the repression of these endogenous transposable elements. And so they have a Crab domain, which normally silences, recruits epigenetic silencing machinery to these transposable elements. And when we again looked at these mutant KRAS lung cells, what we saw again in this volcano plot is that there was a significant downregulation of these Crab zinc finger genes that led to this apparent upregulation of these transposable elements. And we did some other studies, which I don't have time to go into, where we simply took these same cells that had this very significant downregulation of these Crab zinc finger genes, and we reintroduced individual Crab zinc finger genes that were missing to see if we could rescue this phenotype. And indeed, we did see that specific transposable elements were downregulated due to this, this Crab zinc finger gene overexpression. And so we think that these downregulated Crab zinc finger genes are leading kind of to this global epigenetic reprogramming of these cancer cells, leading to this aberrant upregulation of these transposable element RNAs. And again, we wanted to compare this with in vivo data using the Cancer Genome Atlas. And here what I'm showing you is now a comparison between lung cancer primary tumor biopsy RNA-seq data versus RNA sequencing from healthy tissues. So this is lung tissue from healthy individuals. And here you can see, for example, of course, they wouldn't have any mutations in KRAS. And if you compare them with lung cancer patients, lung adenocarcinoma patients with these gain-of-function mutations in KRAS, what you see is that for all of these Crab zinc finger genes, which I just showed you in the previous slide, were significantly downregulated by mutant KRAS, there is this kind of subsequent effect in vivo as well, where when you have this mutation in KRAS, you see this significant downregulation of all of these Crab zinc finger genes, leading to, again, this aberrant upregulation of these non-coding RNAs. And then because we saw this happening in vitro, we really wanted to see what is actually being secreted, and to really be able to diagnose disease non-invasively using these liquid biopsy approaches, we really need to look at not what's happening within the cell, but what's happening outside of the cell in terms of what's secreted by these cancer cells. And so what we did was we looked to see in these cells, again, that we had transformed in a dish, simply with mutant KRAS, what are the RNAs that are being secreted from these cancer cells? And so we isolated these extracellular vesicles, which again kind of protect this RNA that's being secreted. And we performed RNA sequencing to identify these extracellular non-coding RNA signatures of this mutation in KRAS in this very early model of transformation. And what we saw was initially when we characterized the size of these vesicles, here what I'm just showing you is the size distribution of these extracellular vesicles. And this is the diameter of these vesicles in nanometers. What you can see is if you look at control lung cells, which don't have this mutation, typically the size, the diameter is around 200 nanometers. But when you introduce this mutation in KRAS, there is this shift both to slightly larger sized vesicles, but also to slightly smaller sized vesicles as well, what are kind of canonically exosome sized vesicles, which are typically 30 to 150 nanometers in diameter. And then when we looked at the payload of these vesicles that were being secreted by these mutant KRAS cells, what we saw was that indeed there was this preferential secretion of these non-coding RNAs, for example, these long non-coding RNAs or link RNAs, as well as for protein coding RNA, but you don't see as kind of distinct of a bias in terms of what's being secreted. So here on the right is all of the RNAs that are preferentially in these vesicles, and then on the left are the RNAs that are preferentially retained within these cancer cells. And so again, there is this very strong signal that's coming from these non-coding RNAs that we believe will be very powerful for, again, diagnosing disease at the earliest stages. And so here, this is just kind of the model that we favor right now in terms of how this very early mutation, this very kind of early change in the progression of cancer leads to this upregulation of these non-coding RNAs, such as these transposable element RNAs. which are then secreted in these vesicles into the circulation, into blood, which can then be detected via these RNA-based liquid biopsies. So now that we kind of understood what the types of RNAs or the classes of RNAs were that could really be informative for this diagnostic-based approach, we really wanted to then see, can we develop an RNA liquid biopsy platform that enables this cancer early detection, and really to leverage, again, these RNA signals that are being secreted in these vesicles. And based on this, this in vitro study that I just told you about, we really were able to identify these non-coding RNAs as the most salient features that we could use for this early stage cancer detection RNA liquid biopsy platform. And so kind of going back to this, this idea of feature selection for this machine learning based cancer early detection platform, what we wanted to do was, because it's difficult to, for example, if you have like 1 million different RNA features, it's difficult to get like millions of patient samples that would allow us to properly train our model. What we wanted to do was we wanted to reduce this feature space as much as possible. And to do so, we used existing data from the Genotype Tissue Expression Consortium, or GTEx, as well as the Cancer Genome Atlas. And so here, what's really quite nice about these publicly available data sets is that, for example, for the GTEx Consortium, what they've done is they've looked at 54 different tissue types across hundreds of different patients. And so in aggregate, there are over 15,000 patient samples across hundreds of individuals for 54 different tissue types, which are indicated here in this diagram. And so what we can do with this is to identify what are the signals that you would see, for example, in a healthy individual. If you were able to do kind of this RNA-based diagnostic approach, and you were able to sequence, let's say, the blood of healthy individuals, what are the signals that these healthy tissues would contribute to kind of this baseline of healthy RNA information in the circulation? But then also, for example, for cancer, we wanted to see if we use the Cancer Genome Atlas, can we then take, for example, now these RNA sequencing data sets from over 11,000 patients across 33 different tumor types, and to really identify these disease-specific RNA features for individual types of cancers. And the nice thing about this TCGA data set is that, of course, it's very large. There's tons of data, as you can see here, but there's a lot of RNA sequencing data that could then also be matched to, for example, mutational information from these patients. So they've also done DNA sequencing. There's epigenomic profiling of these samples as well. There's imaging as well. So there are a lot of additional kind of data types that you can use to kind of enhance your ability to identify disease-specific features. But just looking again at the RNA, you can identify based on looking at these over 11,000 plus over 15,000 RNA sequencing libraries that were generated from both healthy and cancer, what are these disease-specific RNA features that we can extract? And so I'm just going to give you one example of what we did. And so essentially what we wanted to do was, let's see what is the signal, for example, that's enriched in healthy kidney tissue. And so here what we're doing is we're looking at all of these different samples, for example, across GTEx and TCGA, and we're comparing everything except kidney to kidney. And so we're performing this differential expression analysis here, again, with the volcano plot indicating on the right, all of the RNAs that are significantly enriched in kidney, but not in any other tissue or any other cancer type within this large data set. So what's really interesting is that you can identify RNAs that are significantly enriched within one given tissue compared to everything else. And again, going back to this is across 54 plus 33. So there's like 87 different tissue or cancer types, and then 11,000 plus 15,000. So we're looking at over 26,000 RNA sequencing samples. And so again, when you do this one versus all comparison, you can see that there is indeed this tissue-specific signal for healthy kidney. And the nice thing is, what we also did was we basically, in addition to the standard protein coding genes that everybody tends to look at in these data sets, we wanted to look at the non-coding feature space as well. So we added kind of in this custom annotation of the transcriptome, we added a lot of different non-coding RNAs, including the hundreds of thousands of long non-coding RNAs and these other repetitive RNAs as well. And so what you can see here in red is these non-coding element-derived RNAs. And so indeed, you can see that there are additional kind of signals that are coming from these non-coding parts of the genome, where you can see this in healthy kidney. But what was really quite surprising and striking to us is when we did this now for kidney cancer instead of for healthy kidney, we see a very different signal. And so this is, again, a one versus all comparison where we're looking at what's significantly enriched in kidney cancer RNA sequencing data sets from kidney cancer patients in TCGA. And we're comparing that to all of the other RNA sequencing data sets across GTEx as well as TCGA for non-kidney cancers. And so what you see is there are a lot of signals that are coming from these non-coding parts of the genome. So again, red is non-coding, and black is actually the coding or gen code annotation. And so what we see is, again, that there are a lot of signals that typically are not found in healthy individuals. So going back to this healthy kidney, so we don't see, a ton of that signal, where a lot of that signal is now being aberrantly upregulated in these cancer patients in vivo. And so what was really exciting about this is that now we can leverage this very, tissue-specific, not only tissue, but disease-specific and cancer type specific signal, these disease-specific RNA features that will enable us to do this type of classification to identify whether these patients have cancer or not cancer using this liquid biopsy-based approach. And so what I've really hopefully convinced you of is that this is a really powerful approach for this featurization, where you can now take millions of these RNA features in the genome that include these non-coding RNAs, that include these repetitive sequences, and now we can bring them down to dozens or hundreds of features. And the really powerful thing about this is that in addition to really having that very kind of a smaller feature space, like they have this very strong tissue and cancer kind of specificity in terms of where they're expressed, in terms of the disease specificity. And so now when we're doing these kind of ML-based approaches, we can, instead of using millions of features, we can now use dozens to hundreds of features, and that would greatly kind of reduce the likelihood of overfitting, reduce the need for like millions of samples. Now we can do these types of studies with hundreds to thousands of samples, which is much more tractable. And so we're really excited about this because now we can use these RNA signals based on this one versus all comparison to identify the tissue of origin. So for example, if you were to do this liquid biopsy in the future, let's say that this was standard practice a few years from now, where you can just draw a few milliliters of blood, sequence all of the RNA that's circulating in the blood. Now, when you see specific RNAs, you can attribute them to specific tissues of origin. So for example, for the kidney example, there were these few hundred genes that we saw that were very specific to kidney. So when we see those signals enriched in the blood of, for example, patients that have done this RNA liquid biopsy, we would be able to say that signal is coming likely from the kidney. And if they did have, for example, kidney cancer, then we could then use standard imaging modalities to confirm the presence of, for example, a tumor within the kidney, and then to apply a lot of the exciting work that Olivier is doing, combining imaging with some of these genomic and transcriptomic features as well. And so with that, I'll just end there and say that we're really now extending this to be able to develop a multi-cancer early detection RNA-based liquid biopsy test. Again, leveraging this vast amount of data that we've reanalyzed from TCGA and GTEx to identify these disease-specific RNA features, as well as to do this tissue of origin deconvolution as well of the signal that's coming from the blood. So with that, I just want to thank all the people in my lab that really contributed to this work. Two really talented grad students in the lab, Roman Reggiardo and Andy Davidson, really drove the aspects of the work that I shared today. Really thankful, especially to funding sources from especially the American Cancer Society, who really is really spearheading all of the funding for this work. And we're looking for talented grad students and postdocs, so please reach out if you're interested. Thanks very much. So I guess see if there are any questions in Slido. Okay, so there's a question. Daniel, how much data are involved? Do you learn features from data or design your features? Yeah, that's a great question. So we're looking at 26,000 different data sets, well, libraries, I guess, RNA sequencing libraries. We're looking at, all of the RNA features that I mentioned. So all of the protein coding RNAs, as well as the non-coding RNAs. So we're looking at, upwards of millions of different RNA features within the genome. And what we're doing is we're basically kind of reducing the feature space by doing this one versus all comparison of, for example, kidney cancer versus all the other GTEx and all the other TCGA data that we've looked at to kind of reduce that to a manageable number of features. So we've looked at, for example, like the top 25 RNAs that are significantly enriched in, let's say, a particular cancer or tissue type. And when we compare, the top 25 across all of the different samples that we've looked at, it ends up being about 800 RNAs that have this, cancer-specific information as well as tissue-specific information. And so those are kind of the features that we would use for subsequent kind of classification. Okay, so next question from Jane Yi. Can the RNA liquid biopsy method also be used during and or after treatment as a test, a cure, or a test for efficacy of cancer treatment? Yeah, that's a great question as well. So definitely we see the potential for this platform to be able to use this during treatment to just see the efficacy of the treatment. So for example, I talked about RAS. There are now these very specific KRAS inhibitors that are being developed by several different companies now. And also there's an FDA approved one from Amgen where they're targeting KRAS G12C mutations. And for example, if you were able to identify patients very early on that had, let's say, just this KRAS G12C mutation, and you were able to treat them with this small molecule targeted inhibitor just for that KRAS G12C, that would potentially be much more effective. But you could also see how effective that treatment is by looking for how the signals that are in the blood change due to that inhibition. And so definitely we think that this is potentially useful for monitoring disease efficacy, also for detecting, for example, minimal residual disease. We think that it would be potentially more sensitive compared to DNA as well, because again, the cells don't have to die. The cells are constantly secreting RNA in these vesicles. And we're, again, not looking for a specific mutation in a specific piece of DNA. We're looking at how that specific mutation in that one piece of DNA, that one gene, is really altering this vast transcriptomic landscape of like the 75% of the 3 billion base pairs that are being transcribed. And also, yeah, that would hopefully give us more signal to be able to detect disease recurrence much more early on. Okay, so I think those are all the questions in the Slido. So yeah, thanks everyone.
Great, thanks, Daniel. Let's go on to the final speaker, Dr. Edward Chang. He is founder and CTO of AI, and he's also adjunct professor at the Department of Computer Science at Stanford. And his research interests are in conscious modeling, meta learning, and healthcare.
Thank you, Daniel, and glad to talk to all you guys. And the discussion today is actually focused on some research philosophy of artificial intelligence. So I work on the healthcare subjects for about 10 years and AI for about 20 years. But let's pause a little bit to see, well, in AI applying to healthcare in the last about 10 years, what have we accomplished, right? An interesting measure is life expectancy. If we look at life expectancy, and this data set shows something pretty interesting. And in the US, our life expectancy is actually lower than comparable advanced countries. And another interesting statistic is since about year 2009 and to 2019, about 10 years, the improvement of life expectancy in the US was actually flattened out. And that was actually quite surprising, because if we consider AI to be kind of improving healthcare, and this particular picture shows here, and the life expectancy from 2009 to 2019 was flat. So the question is why? And these are some discussion made in the industry of healthcare So people recognize there is a gridlock between cost, accessibility, and the quality. So if we try to improve one of the factors, let's say we want to reduce the cost, then the quality may reduce. And at the current situation, if we look at the cost in the US, the average spending per person is $12,000. That's a lot. And in the entire world, about 2.5 billion population, they don't have access to healthcare. And in Sub-Sahara and Southeast Asia, the doctor to patient ratio is 1 to 2,000. And the quality, even in the US, the medical spending for mistreatment, misdiagnosis is also very huge. So how do we tackle the problem? Of course, last 10 years, people saying, well, AI, using AI to tackle the problem. But the first question is, what is really AI? Because we talked about AI for 3 generations already, and the last 2 turned out to be a bust. And how about this generation of AI since 2012? And to distinguish this generation of AI versus versus statistics, the only major difference is we have scale of data proven by the AlexNet, and we need to require not only volume of data, but also diversity. And once we have a lot of data, then we require scale of computation. So therefore, like NVIDIA, the stock price took off since 2014. So if you consider difference, key difference is rather than handcrafting a model, and in this era of AI, we are learning models from the data. So about 2 weeks ago, I went to Everest with some colleagues of Stanford, and they talk about geological modeling of global warming. In the last 30 years, all the human crafted models turned out to be not very, not accurate. So currently they are trying to collecting a lot of samples of the world, try to go for this kind of data-driven approach. And about data-driven approach, as if you have attended machine learning conferences, you know the major problem is lacking interpretability, and we have no actually good solution for all the distributed generalization. And what I'm going to talk about today is probably talking about the problems and also some potential solutions in the future. And in about 3 years ago, Yoshio Banjo in the NeurIPS keynote, he was saying, well, maybe we should demand a paradigm shift in AI. So rather than dwelling in the current AI data-driven, so maybe we should go forward to build up system 2, which is developing planning, reasoning, and basically building consciousness on top of unconsciousness. And the current AI is purely computational. You can consider the current AI is just totally unconsciousness. One analogy is AI, if you give the same input, the function returns you the same output. That means the AI doesn't really have consciousness or free will to give you different results. Okay, accessibility is kind of 2 ways. One is patient assessing the healthcare platform, but on the other hand is, and how can we assess data? So this morning, and Professor John Hollenbach talked about Mayo Clinic has this distributed triage platform. I think that's interesting. We also done some work in that direction. And the other thing is, how about data? If we want to collect all the data in the world, and at the same time, we would like to be able to preserve privacy, then what kind of platform we should offer to the public? And I don't have time to talk about this, but welcome to kind of reference the paper, Soterio. And there was a very interesting kind of 3-tier blockchain kind of architecture can address the problems mentioned by John this morning. And of course, we have regulatory problems. And also for healthcare, it's really challenging to deploy because of the business model tend to be B2B and then to C. So every hospital, for example, in Taipei, I work with about 10 different hospitals, but every hospital has its own kind of data format, different operation, kind of procedures. So you cannot just build one system fit all, right? And it's unlike selling cell phones, you just have one specification you sell to everyone. But for healthcare, you have to talk to every single individual hospitals. And then above that, there's regulations, privacy, and also kind of certification policies. So in terms of quality, it's much easier to discuss. I think all the researchers today or before talk about getting feature learned, then improving prediction accuracy, right? So if we want to break the the gridlock, first of all, we want to be able to improving tri-edge kind of timeliness. Because if we delay the treatment, then of course the patient may die. And in about 10 years ago, we attended or participated in a competition fostered by Enterprise. And the competition called Enterprise Tricorder. The idea was in Star Trek, this character Spock wearing a cell phone kind of devices, and that device can do a bio-scanning and also healthcare scanning. So Tricorder Enterprise in about 10 years ago saying, well, if you can develop a very lightweight device which can diagnose some key diseases, and that may be leading to accessibility breakthrough. So there were about 310 teams participated, and we were end up in the top 2. So this is an example of this kind of tricorder. It's a very small box. This was built about 7 years ago when the competition ended. So if today we want to build the same thing, the size will be much smaller. And you open the box, there are multiple compartments. And on the left-hand side, we can conduct blood tests, urine tests. And at the bottom, we do vital sign and the ECG and so on and so forth. On the right-hand side, there are cameras and can do a skin cancer detection and otitis media kind of diagnosis. And the key thing in the middle is kind of called a symptom checker. And this is really similar to tri-edge. Because if you at home, you have this box or you have this box in a nearby CVS, and you don't walk into a hospital, you do every single lab test, right? You want to kind of doing an interaction session and doing some kind of diagnosis. Then at the end, the system suggests you what kind of test you should do. So let's look at this symptom checker. So if a patient at home is not feeling comfortable and he or she knows this box can diagnose the following diseases, then we start a symptom checker process. Basically, just like we go to a hospital, the doctor asks us our symptoms, and based on the first few symptoms, the doctor can ask additional questions. The key thing is to try to ambiguate all the confusions and to hone into the potential diseases. And this is a very challenging problem because the number of symptoms is about 800, and but the number of diseases perhaps 200 plus, and the space is very large. And one challenging part is it's really hard to get, let's say I query you, what is your symptom? But if the symptom I enumerate cannot get a positive response in the beginning, then actually there's no information I can obtain from the patients. Once you get a 1 saying, yes, I have fever, then subsequent 0 means something. But if you don't get a yes, right, you say, oh, I don't have nothing, whatever, then it's really hard to kind of start to do analysis. So this problem can be addressed by reinforcement learning. And I think pretty much everybody knows about reinforcement learning, was utilized by AlphaGo and become the unbeatable champion. And what is the difference between AlphaGo in this gaming scenario and compared to the healthcare scenario? If we can mimic the same algorithm, right, reinforcement learning, if we can just mimic the AlphaGo's training process, yeah, we can do a lot of perfect things. And but the major differences are the following. First of all, AlphaGo, and it has countable discrete actions, right? You have this Go board, and you place the stone on the board. Your input is deterministic. But our body system consists of uncountable continuous attributes. And for a game, outcome is binary. You win or you lose. But the outcome of diagnosis, the outcome can be 1 or a few out of 800 diseases. And in the AlphaGo situation, it can play with itself and exploring all these unknown spaces. And if it loses in the experiment phases, it's not going to kill anyone. But there's no avenue for healthcare to do exploration. You cannot say, well, you are not feeling well, I give you a random medicine to see whether you can get better or not, right? So because healthcare faces the biggest challenge is we are facing small data. And many researchers have talked about transfer learning, dimension reduction, all those methods. Those have been utilized for years. And we started to do transfer learning about 10 years ago. For certain diseases, magically work. For certain diseases, they just don't work. So I will give you one example to discuss. Maybe you can predict transfer learning will work for the scenario or not beforehand. Okay, so if we have a tricorder device, then this traditional kind of medical reactive system, right? You have some kind of uncomfort situation, go to see a doctor, and the doctor saying, well, you should do some lab tests, and after the lab test result comes back, then you do another appointment to see the doctor, then you get your treatment. And this is quite a long process. And if we have a device at home or continuing collecting our physical data, just like Tesla collects billions of data points every day, I mean, collecting our health-related data daily is really reasonable, right? Once we have a lot of data created or collected, then this healthcare cloud, AI cloud, can do some prediction, then sending alert to us if there's any potential problems. At the same time, the healthcare cloud can send information to your physicians, and the physician may say, well, you may really have something, I have concern, so invite the patient to go to the hospital. And before the doctor sees you, already the lab test can be prescribed, so there's no kind of delay situation. And this is proactive and preventive, and because data is abundant, so the diagnosis can be much more precise. Okay, so how can we improve diagnosis accuracy? And this example is very similar to all other examples shown in today's conference. So suppose we want to go for this diagnose otitis media accurately, and then we go to hospital saying, well, we have some samples, you have diseases or you have no diseases, and the data may not be sufficient. In our example, about 10 years ago, we collected about 3,000 kind of pieces of imageries, and they are really in a small data regime. So we put all the data in a CNN, and we get 75% accuracy. It was quite disappointed. And the doctor say, okay, let me tell you what are the appearance or symptoms of otitis media, and maybe bleeding, maybe you have a kind of infections. So the doctor, using domain knowledge, taught us to do segmentation, extracting features. So we were able to improve the accuracy to about 80%, and that's it, right? But we say, well, in the AI era, how come we cannot learn features from the data rather than we have to teach the data by the physicians? So one of the engineer, this is many years ago, today is a common practice. You use ImageNet, whatever, pre-train data, pre-train a model, then you do fine-tuning. At the time, this is 2015, although the paper later was aggregated in 2017. So we use pre-training of ImageNet, accuracy was improved to 91%. The key question is where the improvement comes from. And this picture was in Andrew Ng's early paper. Basically, we have some target semantics, and if we look at the middle layer neurons, you probably can see the parts of the objects. And at the bottom layer are all these kind of fundamental features, edges or contour, whatever, all different semantics can share. So for sure, if we go for transfer learning using ImageNet, we can borrow from the ImageNet all those kind of edges, contour, those fundamental features, all objects can share. But how about the middle layer features, right? So we did some visualization, but again, visualization or interpretability is really kind of application domain specific. There's no a common method right now can do interpretation for all different diseases. But let's look at what we found out. On the left-hand side, this is a normal eardrum. And we look at what kind of neurons in the middle layers got turned on. And we see amoeba or bugs. And on the right-hand side, there are 3 different appearances of otitis media. Then you see a red silk kind of showed up for bleeding, and you have this kind of latte or coffee or orange, half orange shows up for infectious eardrum. And the idea here is actually the neural network is doing explicit stimuli, right? Explicit stimuli is like, well, the ocean is so blue like a sky. And implicit stimuli is like, well, you walk so slow like a turtle. Unfortunately, AI cannot do implicit stimuli today, maybe in the future. But clearly, explicit stimuli can be captured. But then the question is, when you do transfer learning, do your source domain and your new target domain, do they have shareable stimuli? If not, right, for example, we use the same approach, try to apply for detecting ECG diseases, heart diseases, and melanoma. and the improvement is really, really limited because there's no middle layer analogy can be conducted. So one thing I think people can actually work on is you may want to kind of do a prediction whether the target domain can borrow any information from the source domain. Okay, small data. And so everybody is doing the following. Dimension reduction has been done for many years, 20 years, and the data augmentation and transfer representation learning. And also there are people using GAN models to generate more data, and also people using federated learning. But what federated learning is okay for textual data, but not working for heterogeneous imagery, because images may be captured in different hospitals in different resolution using different machines, and the local models, even we all use the same CNN models, the parameter tuning can be different. So you say, well, because privacy, we cut a middle layer, then we aggregate the data, then we aggregate the metadata, and then we kind of train another model to try to train a unified model. That won't work, right? It's really easy to understand. The data is really noisy. And so frankly, we are hitting the wall. The key is without large volume and diversified training data, there's only so much you can do. And whatever you can do is not really falling into the region of the current era of data-driven AI. We are still doing statistic or traditional machine learning. That's not a problem, but we haven't made a lot of progress using the traditional method on small data, and we are not expecting the traditional method can lead to a breakthrough. And also, we have this FDA certification process, and when we develop some applications, we face a challenge of, suppose we have an improved version, then the FDA actually require you to just resubmit the application again, instead of saying, okay, just like any apps, you can upload a new version. And the insurance may not cover the new kind of treatment process. And virtually all the internet IoTs will encounter intellectual properties because all those diagnosis process was already invented years ago, and the patents owned by the big companies. And when you try to miniaturize those kind of technologies, and you may get lawsuit. So GAN doesn't work. Let's do a very simple analogy. So suppose you say, I have collected some red roses, and I like to be able to generalize using GAN to generate other colors of roses, so I can learn the whole category of kind of roses. But there's no way where when you have red roses as your distribution, the GAN will reject other color of roses. This is just the nature of this kind of generating model. And the discriminator will say, well, purple, whatever other colors, it's not in your distribution, original distribution, they are fake, right? So how can we break out this kind of situation? And this another example is like stroke. We know there are different kind of strokes. And in Taipei, we say, well, we use scans to generate additional images, but the CT scans. But the doctor look at us saying, well, whatever you generate, most of them are rubbish because the blood kind of whatever outbreak of the blood is not really on the blood cells. It's kind of randomly distributed on the brain. It's not realistic, right? So you really need to have domain knowledge and to generate realistic data. Another example is for thoracic diseases, and we also try to use scan to generate more data. There are two ways to generate it. One is we just generate data for all categories. And there are 4 different kind of diseases, different kind of lung diseases. Or you say, well, for positive, we have one GANs. For negative, we have another GANs. But at the end, when we compare the accuracy, all of them are worse than using ImageNet as your pre-trained model. That's really ridiculous. You say, well, I based on the lung cancer images to do GANs, but it turned out the results worse than I use ImageNet, those kind of random ImageNet with coffee cups and or vehicles to be the pre-trained model. So there are just a lot of noise in this kind of pre-training, whatever scenarios. So there's not much to be celebrated about. Okay, logically, to break out from the current pit hole, we will say, okay, suppose you want to generate a different color of flowers, a different color of roses. then you probably need to have the knowledge about ROSES. Go to Wikipedia to read about the specification of the ROSES and using the knowledge to guide you to generate new data, right? So guiding is very important. You guiding the generator to explore diversity productively. At the same time, you want to allow the discriminator needs to allow diversity reasonably. Reasonably means I only reject certain things based on knowledge rather than just based on the original distribution of the data. So this is a paper we wrote a couple of years ago, but was not accepted, and I don't need any publication anymore, so we just filed it away. The key critiques from the reviewer was, well, for every single domain, you need to kind of acquire knowledge, and this is not very scalable. I totally agree. So this is the last topic we kind of today want to dive into. Okay, effective treatment. Okay, that's another thing. Effective treatment, let me skip because it's not more critical. So recently, about starting of this year, large pre-trained language model has been developed and deployed, and people started using prompting as the method, and find out prompting is more effective. Effective means require much less data than fine-tuning to get some high accuracy. So the question is, can we utilizing this kind of new paradigm to be able to kind of acquire knowledge from your pre-trained model and using that knowledge to guide your new data, novel data generation? And we think it's possible, and we have done some experiments, and my colleagues in the computer science department, Professor Monica Lam, over the summer, we developed a system to teach the autism patient to speak empathetically, right? So how do you mean empathetically rather than creating thousands and millions of data to teach a computer? And we basically just prompting the computer saying, I'm your good friend. And when I say something like, I got a promotion, you shouldn't say you got a promotion too, because that's one up. You shouldn't say, who cares, no big deal, because that's kind of jealousy or whatever. You should say something like, oh yeah, congratulations, and you must have done some good job. So those are, we call prompting, but so far prompting is in a very chaotic situation. You look at different researches, and a lot of hacking is going on. And Google came out with a chain of thought for mathematical computation. That's very interesting, but there's much work to be done. So basically you have a pre-trained model which has a lot of information, all the information perhaps in the world, and you have so many neurons, so many layers. And then at the same time you say, well, can I use prompting using some sort of like dialogue to acquire data, acquire knowledge from this pre-trained model? And if that can be done, then we can use that pre-trained knowledge, pre-trained model's knowledge, and try to fuse into the generating model to generate novel data. So this was a talk I gave in IEEE infrastructures and also another conference. If you are interested, you can look into my homepage. And this is pretty much what I have today. And I teach AI for healthcare in Stanford, and my last 6 to 8 lecture was try to model consciousness. So this is another topic if you are interested. And it's still not mature yet, but we already been able to model something like love, caring, and the different kind of sins, and even free will. Right? So it's an interesting topic you can look into, and it's really motivated by Yoshi Banjo, considering system 1 is at the end of its kind of futility, and we have to consider system 2. Oh, thank you. Thank again to all my colleagues, and also I put some references at the end. Okay, that's my talk, and see if you have any questions. Okay, if we don't have questions for my section, then my session is concluded here, and Olivia, you can take over.
Great. I still, if anyone still has a burning question for Edward, please submit it online. We have a few more minutes. So there is a question, can you talk more about augmenting the data sets?
So data augmentation was performed by AlexNet in the beginning, right? They do rotation scaling of the original images to try to create a lot of different varieties. And the fundamental reason is because if we look at an image which is scaled, cropped, or downsampled, we will see the processed image is very similar to the original one. But if you use any kind of distance function, the distance function will tell those two images are totally different. So doing this kind of augmentation is trying to you imagine all different kind of transformation on your original image, and you try to defend that. But there's no way you can do for all, right? So recently, that graph neural net is saying, okay, if you have a graph structure, then you can deal with certain transformation in the prior, right? For example, you scale, then translation is dealt with. But the graph neural net only can deal with 3 to 4 kind of transformations, not all different transformations. So how to do data augmentation itself is, again, a black magic, right? So it's really tough.
There's another question. Do these techniques lead to unintended biases?
We don't know, but I think at least the new method we are thinking about doing, modeling consciousness, the basic difference is for computational model of AI, or you call distributed AI, it's like a function, right? Different people give the model or whatever, the same input, you get the same output. But if you model consciousness into your framework, then it's no longer a function. And if the response is like crazy out of control, then that could be hazard. So therefore, we have to put into guardrails to try to make sure the actions of the machine in their consciousness mode is not crazy as human. Like human can commit crimes. So one example is we model sins, different kind of sins, try to prevent the robot to commit crimes into the consciousness.
Great.
It's a little bit far-fetching, but I think we are getting there in about 2 or 3 years. This will become a popular topic.
Great. I think we can wrap it up here. Thanks, Edward and Daniel, and thanks to the conference organizers for putting together this session. And thank you to the audience for attending. Hope to see you at another time.
Thank you. Bye-bye.